All right, folks, so we're going to start looking at our rat dissection. So first things first, if your rat is in a bag, you're going to need to take it out. If it's not, that's okay. So we're going to go ahead and pull our rat friend out and just put him down onto your dissection tray. So I'm saying him, but this one happens to be a female, you know, because you don't see any testicles here. And so there's a few things we need to do to orient ourselves before we get started here. So I told you at the beginning of this class that we are not going to be using the terms ventral and dorsal on us as humans in our human biology class. Instead, we're using the terms anterior and posterior. The main reason for that is anterior and posterior and ventral and dorsal actually mean different things. In humans, they seem to mean the same thing, but they don't. So ventral is belly side. Anterior is like face side. So in humans, our bellies and our faces are on the same side, but in four-legged animals like these rats here, they are not. So what you're going to be doing here is this portion, the back portion, is the dorsal side. So you're going to put your rat so the dorsal side is down and the ventral side faces up, okay? Now we're doing this because most of the dissection stuff we need to do on the ventral surface, all right? Now, some people may have rats that already are partially dissected in this area, and some people may not. Every semester it's a little bit different, and it depends on who we bought the rats from and what was previously done. Okay, this one happens to already have some of the dissection done to the skin here. That's okay. But essentially, for today, we are going to be doing the respiratory system. Okay, so remember the respiratory system is in the thoracic cavity. So it's going to be right up here on front. So, few things that we need to know before we actually get started. First of all, I want everyone to find your probe, okay? Your probe you should find in your dissection kit. It's best if it has a blunt end. There are ones that have pokey ends, but those ones are going to stab things in here that we don't really want stabbed. So a blunt-ended probe is super important because this allows you to point to different structures. You could use your finger, but as you can see, your finger is a lot larger than the tip of the probe, and so things can get confused. Okay, so it's always best to have your probe to be able to point to things. Also, certain things you're going to need to use your probe to get under, which your fingers can't do. All right. Now, other important things that we're going to need. You are going to need a scalpel. All right. Now, the scalpel essentially is only going to be used in certain situations. I don't want you to try to use it for other things. Okay. We'll talk about when to use it and when not. You're also going to need to use your scissors. Okay. The scissors is going to be helpful in certain things where your scalpel is not. All right. Now, these tools can be just put directly into your dissection tray. That's perfectly fine. At certain times, we may also need to use some of our T-pins. These T-pins are helping to hold things open, okay? You can stick them directly into here and they will hold in place, okay? And so some of those may be needed at certain times. Other than that, the other things in your dissection kit you probably won't need, okay? So these are our main items, all right? All right, folks, so we're going to go ahead and start the respiratory system dissection. And you'll notice this is a different rat. So I got a different one that isn't already partially dissected so that we'll have a good idea no matter what. And so still he is laying on his dorsal aspect. So the first thing we got to do, take his little arms, move them straight out. Okay, you heard those little crunchy sounds? That was me breaking his arms. Don't worry, he didn't feel it, okay? But we need his arms spread out a little bit more. And so this gives us a good view of this whole section. So you take your scalpel And when you're using a scalpel, you always want to cut away from yourself. This way, if it slips, it's not going to hit your fingers. So we're going to press it lightly around his throat, and then we're going to make a small cut. Okay, I say small because once we have that small cut, we are going to use our scissors, get inside the cut, and then extend the cut down. Okay, now what I'm going to do right now is I'm going to actually flip him because I'm a little too right-handed to be able to complete this as is. Okay, so you see here how I now have the scissors into that original cut, and so I am just going to use the scissors to extend the cut downwards, and we're slowly cutting open his skin until we get to about there. Now, you can see that this is his axillary region, otherwise known as his armpit, and we're going to about there. So we're a little bit below that, and then from there, we're going to extend the cut laterally until we get to about the end so that we're equal with his axillary region. And then I'm going to flip him around and do the same thing on the other side. So again, we're about even with the axillary region, okay? And so we've now made a flap, and we can start to pull up this flap. Now you'll notice it's still attached, so you're going to pull carefully, pushing down on the muscle, and that is going to separate the skin from the muscle underneath. Okay? So we're pulling it out and carefully separating. So we have skin on one side, muscle still attached over here. Okay? We should only have cut through the skin. Now, I'm going to extend the cut up here by his throat with my scissors again. And this is going to allow it to again pull out so that we have now a square region. Okay? So this square region, we can now use our T-pin. And we're going to push it through the skin and hold it in place on one side. Okay? So I'm going to go ahead and do the other side and then be right back. Alright folks, so we have opened up the skin now on both sides, so I'm going to go ahead and grab my scalpel again, and I'm just going to press it in carefully, and once I get in, I'm going to immediately go right back out and switch back to my scissors. Okay? And so I'm going to cut through, and then I'm going to mirror the cuts I did on the skin and cut across this way. And when I pull up, we're going to be taking the muscle and the ribs. So we see that we're now inside the thoracic cavity, and you can see the lung tissue inside. So I'm going to go ahead and continue those cuts, and I'll be back in a sec. Alright folks, so we have now opened up the thoracic cavity, and we are now seeing the organs inside. I do want to point out, you see we have pushed back the muscles and the rib cage and attached them to each of the T-pins. You'll notice there's these yellow lines going through there. Those are his little ribs. Okay, and so we essentially just cut straight through his bones. All right, so in the side of your thoracic cavity, you should see a couple different organs, specifically the lungs and the heart. So this over here is going to be his right lung, and this over here is going to be his left lung, and then in the center you should find his heart. Okay, so at the bottom of the thoracic cavity, you should find that there is a membrane, and that membrane is going to be your diaphragm. Okay? Remember, the diaphragm separates the thoracic cavity from the abdominal cavity. Okay? So we're going to kind of explore a little bit here. So we're going to just kind of lightly push up, and this one happens to be quite friable. Friable means that it kind of just falls to pieces. So that's a chunk of his lung that just fell apart as I was touching it with my probe. That probably means this particular rat is quite old, to be honest, and so he's not doing so well, okay? But we did just lose a piece of it here. But we do see that this lung has one lobe here and another separate lobe over there. Okay, you see how they are separated from one another, because remember, the lobes on the right side there are 3 lobes, and on the left there are 2. Okay, so we're going to lightly push up here, and we can see the bottom or the base of the lung. Okay, now what I'm going to go ahead and do is pushing this back a little bit. I'm going to pull up on the heart. Okay, you see the heart there in the middle? And so what we're going to do next is I'm going to go ahead and grab it just with my fingers like this and lightly pull up. And that was me ripping the heart out of our rat with my bare fingers. Okay, not hard at all. All right, so this is the rat's heart. Now, it just looks like a piece of dirt, quite honestly, or maybe a bean, but that is his heart, okay? Now, what I'm going to go ahead and do very carefully is I'm going to put the heart down onto our dissection tray here. I'm going to put my scalpel, and I am just going to cut straight through the heart, okay? Cutting straight down, and then I'm just going to open it up. And we can see that it has little chambers. Just like the sheep heart that we dissected, we have a left ventricle and a right ventricle. You see that thick wall with the left and very thin with the right. Okay? It has all the same chambers. You can even sort of see some of that rough tissue that would go inside of the ventricle. We call that the trabeculae carnae. All of the same features are inside of the rat heart that were inside the sheep heart. They're just in miniature. Okay? So we've already looked at that in detail, but it's important to realize that rats are mammals, just like sheep are mammals, just like humans are mammals. So all of us have the same shape to our hearts. It's just different sizes. Okay? So let's go ahead and continue here with our lungs. And we can see here better some of those lobes. Okay? So on the right side here, here's 1 lobe, 2 lobes, and back here further is a 3rd lobe. Okay? We see all 3 now that we've gotten the heart out of the way. On this other side here, I think we just lost a chunk of 1 lobe, but we have another lobe over here. We can see that really nicely once we get the heart out of the way and I kind of pull it up. So that is also a lobe of the lung. Okay? So what I'm going to go ahead and do is I'm going to carefully, with my fingers, pull out that lung. And now we get a better sense of what it's like. Okay? It has a smooth surface. We should just squeeze it a little bit. It's very light, very squishy. Okay? And you can see this is the vessels that was bringing the blood to and from the lungs that are broken off there. Okay? So, essentially, those are the structures that we need to find today for our respiratory dissection. We will be working with the rat again next time, and we're going to continue down and do an abdominal dissection and look at some more structures, and we'll spend a little time looking at a few things at that point. All right, see you next time.